So in the last video we looked at DNA structure, we looked at nucleic acids, how they bond together, Watson and Crick, and we then talked a little bit about how DNA is set up, double-stranded, double helix, etc., etc. DNA is very, very well suited to its job, and that is obviously storing, expressing genetic information. So a few reasons why it's so well suited. Well, the bases are actually very well protected from any damage. So they're obviously inside this sort of ladder-like structure, A, T, C, G, T, A, etc. And they're very well protected. So this makes DNA is very stable. It's a very stable molecule, and it's not easily damaged as well. So not damaged. There's 4 different bases in there. They can appear in any order. So it's a little bit like a 4-letter alphabet. DNA molecule is very long. It's a long molecule, and it can store lots and lots of information. There's about 3 billion base pairs in humans. And you've also got the 2 complementary strands. And they can copy the information, repair, things like that. So, if we move on then to DNA replication, little picture here, we'll just talk it through. So you've got your original DNA double helix over here. It will start to unwind at a place called the replication origin. That's where it starts to unwind, and that enzyme is called DNA helicase. Basically unzips, if you like, the double helix. It breaks the hydrogen bonds that are between the bases. Breaks the hydrogen bonds. Then the 2 new strands. So if we got over here, you can see over here and over here, we've got 3 nucleotides that are being added to the old strand. So A pairs of T, C pairs of G. So they come along, they bind on there. These are just sort of free floating in the nucleoplasm. We obviously need a sugar phosphate backbone. So what will happen is an enzyme called DNA polymerase will come along, and it will bind the new nucleotides to each other using these strong phosphodiester bonds, and that will link the phosphate and the sugar on each molecule. So, like we talked about before, linking these 2 structures together. Another enzyme then is going to come along and wind the molecule up, so down here and down here, into a double helix, and you essentially have 2 new strands of DNA. The 2 copies of the original DNA, identical unless you have any mutation happening in there. This is what's known as semi-conservative replication, because you're conserving half of the DNA, or you're conserving. There's not an old strand. There's an old strand and a new strand. Sorry, but there's not half from one, half from others. Old strand, new strand, exactly the same as the original DNA double helix up there. Okay.